When it comes to science, we often picture an old professor droning on with endless details in a very monotonous voice. But what if I told you that communicating science can be both entertaining and scientifically accurate?
Ideally, they would be surprised how interesting this topic is. So, the trick is always I try a bit to hook them in with a little bit of humor, and then maybe they stay because not just is it funny, but they notice that this is a super interesting topic.
We've seen during the pandemic how crucial clear communication is, especially for complex issues like disease spreading or vaccinations. A well-informed public is the bedrock of a free society, but how do we balance making science accessible with staying true to the facts? And in a time where misinformation is rampant and political distrust is spreading, how can we rebuild public trust in science?
That's the even harder problem, that probably if we want to really make science education better or more relevant, is that we have to somehow build up again the trust in institutions.
In this episode, we will explore how effective science communication can debunk myths, strengthen democracy, and help us facing growing political radicalization. How will be the future of science communication adapting to these challenges? Let's find out. Have fun and enjoy our conversation.
Daily dose of honest science.
Hello guys, and welcome back to Daily Dose of Honest Science. I'm your host, Dr. Chani Kumar, and of course you are here for the best medical science podcast that you can hear on any podcast. And today I'm joined by a beautiful guest here. He did his bachelor's in biology at the University of Vienna with a specialization in microbiology and genetics. He did his master's then in molecular biology at the Institute of Molecular Biotechnology in Vienna, and then later on did his PhD at the Center for Molecular Medicine in Vienna in a group that was specialized on DNA repair and genomic instability. Now he's become a science communicator. He is also an author, and he got numerous prizes and is part of the Science Busters. Just some few awards for you. So the Humanitätspreis des österreichischen Roten Kreuzes for amazing humanity engagement during the corona pandemics, or even some Kardinal Innitzer Würdigungspreis für wissenschaftlich fundierte Publizistik. And he's here today with us. So a warm welcome and a big applause for Dr. Martin Moder. Hello, Martin.
Bonjour. What's up? Hi.
What's
nice
up?
To be here.
I have to say it's, I think, the first time for you in English, right? So usually you do everything in German.
Yes, I'm crazy nervous. I'm hoping for the AI to just translate everything I ever produced perfectly into English at some point. So usually I don't even try.
Ah, okay. But you should try.
I should try.
You should try, because the humor that he has, I think everyone would enjoy even in the English setting. So I think we just start right away, dear Martin. So your background is molecular biology, so hi, we are colleagues. Of course, with a great focus on medical applications. And I think the most obvious one to know is, how did you actually get into science?
I don't really know. I think it was an accident. I guess I got somehow primed by my family. My father is or was until his pension professor for statistics. My sister is a physicist. So it was quite present at home, this whole science thingy. Though I didn't intend to become a scientist most of my time, at least while in school. I wanted to be a police officer. But at some point I noticed that maybe, I'm not so much into strict hierarchies and so on. So I was a bit lost, and then I ended up studying biology. I inscribed for the study, and in the beginning it was a bit okay-ish. I didn't dislike it, but it wasn't super exciting. But I figured the farther I got in the studies, from semester to semester, it became more interesting. The more it went into genetics and medicine and genetics in medicine. And at some point you start, altering genes of animals on your computer, and it's super weird. And you notice that this is perhaps the most exciting time to be in genetics ever. And then I stuck to it. I really liked it.
What part was the most interesting for you? So designing primers to just...
the primers, designing. I wouldn't say no. Doing PCR is basically the least interesting thing. I don't have to tell you.
Because it didn't work, or...
the first 5 PCRs are okay, but the 700 after that are a bit terrible.
I always hear that.
I had a massive primer problem at some point. I think I've lost half a year of my PhD studies due to a PCR issue. This is
really
I didn't expect it. Yes.
Wow. Okay.
Anyways, the most exciting thing is hard to tell. I guess the most exciting thing is always the moment where you figure something out, and you're the first one now who knows this. The moment you analyze your data. But, in 10 years of study, there was one of those moments. But it was a nice one. But I think that the first moment that really excited me was when I did my master's, and I was doing brain tumor research on fruit flies. And at some point I had to alter these flies genetically. We did RNA interference, and we had to insert a construct into the Drosophila. And I was sitting there on my PC, and I got this list of gene sequences that we wanted to insert into the fruit flies. Yeah. And I had to manually type them into a file. And I noticed that, okay, I'm sitting here, I'm writing on my computer, AGTGAC, knowing that within a few weeks, this will be the genetic sequence of a living animal. This will be a fruit fly running around, flying around, having a gene that I'm now at this very moment typing on my laptop. And I thought that is crazy. That would have been so crazy 10 years ago. And then when I did my doctor's, CRISPR just came out, the gene scissors, that made it so easy and cheap to alter human genes. And that was also very fascinating to notice that you're a geneticist right at the moment where the question whether we can alter the human genome is changing from being a question of whether we can to a question whether we should or we want to.
Yes.
So I think these were some of the driving factors for my studies.
Cool. For me, it's kind of similar, but I'm more on the biochemistry side, so I'm always amazed if I can recombinantly have a human protein in my tube. That's also something sometimes, or if you do like microscopy, that you see the protein that you tagged, and what does it do in the overexpression. Of course, I did some research on you, and I wanted to highlight highlighted your dissertation. So just here the abstract, because I really like what you worked on, and you even published, right? And I think it was Nature Communications. So the continuous repair of DNA damage is a prerequisite for the maintenance of genome integrity. Therefore, cells have evolved a complex network of DNA damage repair to deal with diverse forms of genomic lesions. Defects in those mechanisms frequently lead to diseases associated with cancer, susceptibility, including Fanconi anemia. Using a genome-wide CRISPR knockout library as well as insertional mutagenesis, we identified synthetic viable interactions in human haploid cells deficient for Fanconi anemia complementation group C. Here we show that the phenotype cells can be suppressed by additional loss of BLM helicase complex, so it's getting now very complex. We demonstrate that this synthetic viable effect is not specific to these cells by confirming this interaction in cells deficient for FA complementation group 1 and D2 that functions downstream of the FA complex. These thesis demonstrates that systematic genomic-wide screening can be used to identify genetic-synthetic viable interactions for defects in the DNA damage response. When I read this, the first question that I want to ask you is, can we explain maybe to the audience what you actually did? And do you know, or can you explain what Fanconi anemia is? And CRISPR, we already said it's a gene scissor, but just that you know, get the audience hooked in, why this study is so cool that you did.
Sure. So our cells, or the DNA in our cells, it constantly gets damaged all of the time. There are estimates that every cell in our body gets, I think it's around 100,000 DNA lesions every day. Mm-hmm. And most of them are repaired. We constantly repair them. And we have several repair pathways that allow those lesions to be repaired. And one of them is the Fanconi anemia pathway. Basically, it's responsible for one kind of DNA damage when you have the DNA double strand. Then there is a kind of lesion that is called interstrand crosslink. It's just when they are chemically hooked to one another and then they don't really separate well and so on, and then they break and all the jazz. So it's a really toxic kind of lesion. And there are people who have a defect in this repair pathway. And then they express the disease that is called Fanconi anemia. And when they have this defect, those lesions do not get repaired. Those people have a very high mutation rate, therefore. So they usually die of cancer, or they develop cancer around their 30th year of life. If they don't die of anemia beforehand, because that's also one of the symptoms. But this is, kind of treatable. So we wanted to know, when people have these lesions, and it's not easy to repair a genetic defect in a human, but it's always easier to destroy something than to repair something, as everyone knows from experience, probably.
Yeah. So
we thought maybe, the whole genome, it's a massive interaction network. Maybe if there's a lesion here, maybe there's a defect in Fanconi anemia pathway, and it's interconnected with everything else in the genome somehow over some knots. Maybe we can take out some other factors so that the whole system behaves more akin to a healthy cell. So maybe we can insert a second defect and everything works better afterwards. Mm-hmm. It's a fishing expedition, what people usually don't like when you write grants, proposals. But it did work, because this is what is called a synthetic viable interaction, as you mentioned. So the cell has a problem if there's just this defect, but if we insert another defect and it works better, then we call this a synthetic viable interaction, because now they're viable, they can live. So we actually took then two approaches. Basically, maybe I shouldn't go into too much depth. But so what we wanted to know, we take these cells and then we delete another gene. And then we test, do these cells behave more healthy now? Or can they live with this form of DNA damage better? And then we delete a different gene in another cell. So we only delete this gene, then we only delete this gene. And this sounds like a terribly boring, long kind of work. But at the end of the day, we have CRISPR, we have vector virus libraries. So we could do this all in a pooled approach. Oh. And we could do this all at once, basically. And what we found is that if you have this BLM complex, as you called it, or most people call it the Bloom complex, if you take this one out, then the cells, they behave better in the presence or let's say after induction of DNA crosslinks, interstrand crosslinks. So that summarizes it. So we did it with CRISPR, we did it with a haploid cell type, so we get rid of biases. And so, you do all this 1,000 redundant stuff to make the reviewers happy.
True. But what I was missing, I think, in the paper, but you can correct me. I think you only did genomics, right? You were not doing proteomics, or?
Yeah, of course, that's what you say. I wasn't doing proteomics. That's true. Yeah. Well, we didn't do proteomics, but I basically identified the Bloom helicase complex by looking at predicted and known protein-protein interactions.
Okay.
So we didn't pull it down, but it was the first time that I noticed that there's a whole complex in my data set is when I was looking at the protein interactions.
See?
But that would have been something, if I would have want to extend my PhD studies for a few years.
Or we
maybe
would
I
have met before, then I would have done it for you. That's why I find it so fascinating to work with other people, because proteomics can give you such a interesting insight on what is happening on the protein level, right? Because when we think of genetics, we have to think of, okay, we have a DNA that is going to be transcribed to an RNA and then translated to a protein. And in the end, the protein is actually doing the work in the cell, right? So I was just wondering why this was not, but I get the point. You cannot extend your PhD to again another years.
I could, but then again, I'm a bit lazy at times. So when I have enough for nature communications, then let's go.
Yeah. So thank you so much for your insights, and it's really amazing. Is there a follow-up to this? I think the group where you worked in is not there anymore, so they moved on. But I would assume that this is a cool target to, pursue for medicine.
I have to say, I'm not sure if there's a follow-up. If there is, then it's not published yet, because then probably they would have cited me, and I guess then I would get a notification somewhere.
Yeah, if they're
I hope they would have cited me, those bastards.
Yeah, no, but I mean, do you sometimes follow up the work that you did?
Me myself.
Yeah. So what is
new?
The truth
is no. I'm,
okay. So you just did it. You're fine. You got your doctor.
I don't think I've ever read a Fanconi anemia paper afterwards.
Oh, okay.
And it's different, if I would have stayed in science in the lab itself, and if I would do my postdoc now and keep sitting on this field, of course, then I would have again a few more years of reading Fanconi anemia papers. But since I've decided to go all in science communication, there is a never-ending list of papers that I want to read. And Fanconi anemia is not the highest priority on my list. Also, I tend to get bored of one topic if I have to exclusively read up on that for several years.
Me too.
So I'm quite happy that.
No.
The highest respect for people who can work on one phosphorylation site for 8 years. I don't know how they do it.
Yes, I also don't know, or they just focus on one pathway and do it in very detail. But for me, it also gets kind of boring, and I also want to know what kind of implication does it has maybe for medicine. So I would like to have this outlook, am I working on something that could change something, and not...
You can always, at the end of the grant proposal, add this might be relevant for cancer, because everything at some point is relevant for cancer.
Well, I'm that far, so I wrote a little fellowship right now, and I'm actually working with a medical professional who's working on multiple myeloma, and it's not made up, so I really have an interest to, decipher what is going on there. But I completely agree that it's not that of an easy thing reading up all the time the same paper, even though I'm from the centromere field. So at some point it gets a bit too much for me. So I'm like, okay, good. This phosphosite, okay.
I see it in my dreams.
Yeah. I just want to hop on your interest, because just some weeks ago we had the Nobel Prizes given, right? So it's kind of cool to see, for instance, the Nobel Prize in Medicine and Physiology went for microRNA and its role in post-transcriptional gene regulation, or in chemistry for AlphaFold, but also for enzyme engineering, and also in general for learning AI and neuronal networks. So I wanted to know from you, what are your kind of thoughts about the prizes that won this year?
I didn't follow it too closely. I was, of course, very happy about the microRNA. I used those to knock down genes a while ago in C. elegans. And I didn't know that this wasn't awarded a Nobel Prize yet. It's quite cool.
Yeah.
But then this year, I was very I thought it's very curious to see that, in two natural science areas, in physics and in chemistry, the Nobel Prize is awarded to basically programmers.
Yeah.
To AI.
Yes.
It's all AI. Even
Yes.
chemistry and physics is AI now.
Yeah.
And this AlphaFold that got the Nobel Prize for chemistry,
Yeah.
it's super exciting, especially for. Maybe we should. I actually don't know who your audience is. Is it people that are in science themselves?
It's mixed. So I have people who are generally the general public who wants to know, but also people like colleagues who are interested in knowing you, for instance. Yeah.
All right, then maybe quickly summarize this AlphaFold, what it's doing. You always had this map because I'm aware that you are aware of all of this, and I don't want to bore you.
I also used it, so
really?
yes.
Yeah, of course.
I'm also having structural biology through cross-linking mass spec, so that's why I used it. Yeah.
Cool. I would like to see how this works in practice. Well, anyway,
we can talk about it in another time.
It allows you to basically predict from an amino acid sequence, predict the structure of a protein. And that is a huge deal because there was sometimes years of work before we could do this on a machine. And what I find so interesting is this guy, I think it was developed by
Google DeepMind,
right?
Yeah.
And one of these guys of Google DeepMind, I forgot his name, he's now the head of a just recently announced new institute in Vienna. That is, I think it's called Etheria or something in that direction. And its mission is basically to use AI in genetics. I was listening to his talk when there was an opening ceremony. And it was so interesting. He had this slide where he mentioned a phrase that leaves me awake at night.
Really?
He was talking about the end of the hypothesis-driven science.
Yes, I heard about this. Yeah.
Yeah, it was mentioned in articles before. And the idea is these AIs, they can work with so much data, much more than we could ever grasp or understand or think in complex networks. So usually you have this one moment where you have to dumb things really down, and that's at least when the machine is spitting out data in a way that you can read. That's the moment where things have to get really simple because we're so incredibly stupid with a tiny brain compared to a massive machine. So the idea is you don't even have to have a great hypothesis. You don't basically you just do a ton of massive screening, whole genome screens, proteomic screens in all different, I don't know, with all different alterations, whatever. And then you take all those data and you feed it into the machine.
Yeah.
And you say, what is interesting? Is there any hypothesis? What can you read out of this? I don't want to, say something about that institute that might be a bit overdone, but that's basically it. We will understand which kind of data we feed in. We might understand the hypothesis or whatever comes out, but everything in between will remain a big black box. And And I guess that might really be how genetics might be done in the near future most effectively.
So you think AI is going to rule and we don't need scientists anymore? Or do we need us?
I'm sure we will need scientists, but I don't know.
To which extent?
I don't know if we will fully. I don't think we will understand how we got to this knowledge, maybe.
Yeah, maybe.
And yeah.
Yeah. And also, if I think of all the people who are now PIs and professors, they are driven by a vision, right? So an AI doesn't have a vision in that sense. So it might change also the landscape of research itself. I would think so.
Yeah, and I wonder if we're missing the important steps in between, because
Could be.
do you want to start? Do you want to have someone as a practicant? How do you say?
Like a Hiwi or like a practical student.
Yeah, practical student which has to learn everything from bottom up, and it's a ton of work to teach them. Yeah. So that at some point they can help you with the project. You know, when an AI can do it better right away.
True. Yeah.
So who wants to teach me if I'm super incompetent at the very beginning? I don't know. I think those are interesting times.
Yeah, very interesting, and so I think it will change the landscape how it is right now. So we will definitely use much more AI, hopefully in a good sense, not in a bad sense. So we covered already a lot, and now I want to go to the next section because it's kind of interesting that you became a science communicator. So now we learned that, he was into science and what made him into science, even though it was not really in that sense. And now I would like to introduce to the audience your current role as a science communicator. And the first question comes, why did you leave academia? Because I think you would have been a great postdoc, even a PI, to be honest, with your, I could laugh all the time. So we would need you, actually.
I don't know if that makes up the best PIs, you know, that they're cracking jokes or something.
At least you would have the humor that some people are missing. Let's say it like that.
So I never had the thought, oh boy, I really want to leave academia. It was more like, besides my studies for a long time, I was working as a tutor in a visitors laboratory where people, you know, could do their own experiments, and you were teaching them first theory, and it was quite advanced at times. We did PCR with school classes and gel electrophoresis and whatnot, and tested their own genes for some characteristics. And I just noticed over the years, and probably far more than 100 different classes that I was teaching there, that it's really satisfying and nice if you explain something to people that is very interesting to you, and then you notice that they find this very interesting as well now.
Yeah.
So I started to do more of this besides my studies. I was doing science slams, and then I started to write my first popular science book. And I just did it, you know, in my spare time. That was my one hobby at the time. And
just quickly, how did you come to the conclusion, I want to write a book? It's something like, you know, like you were just in the lab and this is like a side project for you, and then you decide, I want to write a book about it, or what was the thought process behind it?
Yeah. It's like it's often the case with me that there was not much thought process. It was more the inability to say no. That's a big issue. What I wanted to do was these science slams. So people, sometimes they know poetry slam.
Yes, yeah.
And science slam is very similar. People talk about their own research. It must be their own research. and they have 10 minutes mostly, if at all, and it should be somehow entertaining. And I just wanted to do this once, so I dressed up as Drosophila, as fruit fly, and made one of those science slams in Graz, was it? And I won it. I didn't expect it, but I won it. So they've sent me to the Austrian finals, which I didn't know. I didn't know that there's somewhere where they send you. I just thought you get, I don't know.
You get a prize and that's it.
You get a prize or food or whatever. So they sent me to the Austrian finals, and then I won that as well. And then I got super nervous, but because then I noticed they will send me to the European finals, which were in Copenhagen.
Wow.
So they sent me to the European finals, and then I won that too. And then suddenly I was the first European Science Slam champion. And now the book comes into play, because there was a German Science Slam champion a few years prior.
You have,
and
I don't know.
she...
Yeah. I don't know her, so...
Her name is, maybe you know her because she wrote a super famous book afterwards that became a massive bestseller, millions of copies. It was called Darm mit Charme. So colon with charm. Is this a word? Anyways, it's a popular science book. It got a mega bestseller.
Yeah.
She got super rich. The publisher got a lot of money. So an Austrian publisher thought, okay, this guy, he's not only the local Science Slam champion, he's the first European one. Maybe this guy can write books too. So they asked me, do you want to write a book for us? I said, why not? Why not? You don't get asked from a good large publisher all the time.
Yeah.
Usually people, they have to fight a lot to publish a book. So I said, let's give it a try. And it worked. It didn't get a bestseller, not at all.
I think it is, because it's, it's there and people like it.
It's there. Cool.
And you wrote even a second book, so...
Yeah, I wrote a second book then. And now, occasionally I'm in this science comedy group now, Science Busters.
Yes,
we write books every few years. And actually, thanks for the reminder, I should really write a bit more on that book.
Okay, good. The book will be out in...
I have no idea. There are deadlines, but I try to not think about, because they're
okay. I will write
crazy
you every week a message. Martin, write your book, please.
Thank you very much.
Really cool, and I didn't know that you went through all these little stages, and you're now the European champion. Were there any follow-ups? Like is there every year like a Science Slam coming?
They are all the time. I guess there's still these local and European championships.
Okay.
But I think I never really did an official Science Slam ever after, because, what should come after the European one? There is no global at this point. So
maybe you initiate the global one. Who knows?
Maybe. But... I just don't like giving the same talks over and over, so I basically then just
you could start doing Science Slam about science communication. It's also kind of
counts. Maybe
but it's kind of your research in that sense, right? So
maybe I can publish. Maybe I can call my videos, my YouTube videos, publications. Basically, you
theoretically, yes. Theoretically, yes. Yeah. Cool. Okay, so you didn't want to stay in academia in that sense. You did more of science communication. But what I want to know from you is, based on the experience that you had during your time in academia, what do you think has to change in the research system? Because we have the problem that people are leaving, right? And I think you heard that quite often from your colleagues or people who were in the lab. So what do you think needs to change that we pull them back?
Oh, that's a tough one. I haven't spent much thought on this. It wasn't for me that I wanted to leave. It was just that I noticed that if I do all this science communication stuff that I also enjoy, besides doing my PhD and everything, then I will die very young. So basically I had to decide I should do one of those two all the time. And it wasn't an obvious decision for me. So I was thinking about this a lot, but then I thought there's so many so many good scientists in Austria, and there's so much good science, but there wasn't so much good science communication. So I thought maybe this is the better niche at this point. But it's hard to say what pushes people out of academia from my perspective. I enjoyed it, but of course I also had the outlook of never really knowing if you will be in the same city 8 years in the future, and that's one point of decision, of course. But I don't know if that's something that people should be getting rid of. I know, I could list all these classic. You have to publish or perish all the time, but I'm not well informed enough to really have a vision of a better system. So.
No, I was just thinking like you were in the system, what would have made you stay? But if it was a conscious decision from your side, that you say, hey, science communication is something that I really want to do. Maybe on that note, do you miss sometimes being in the lab, or are you kind of happy?
Yeah.
Yeah?
Yeah, there are several things that I miss. First, I miss working with the same people every day, knowing those are people that I don't only I like, but also being surrounded by super smart people. Now my days mostly I sit here of the day, or maybe in a cafe and write and read, and I just miss being around by people that I consider geniuses. That's really.
You are also considered as a genius, by the way, no? Also,
yeah, but
don't make yourself
people,
so low.
Yeah, but by people who don't know me very well. Yeah, this is one thing I miss, and then just the simple act of having a place where I have to go every day to work. Having to get up at a certain time.
Also planning experiments, right? And how to do things, what could be a really cool experiment to show something, right? Collaborations.
Yeah, I didn't enjoy the planning part so much because it's so confusing. So much. I like the stupid stuff, I like sitting in the cell culture all day, pipetting, and talking to colleagues.
Okay.
I like the planning too, but I'm much more a data analysis guy than I'm a planning guy. So when the data is here, then I have fun looking at them.
So more in the bioinformatics. So you're more
maybe,
but bioinformatics.
Yeah.
Okay. For me, it's kind of the same. So right now I'm a postdoc in the lab and enjoying every bit of it. But of course, I started my journey just a year ago. So you're inspiring me to continue. But I also cannot imagine myself not being in the lab and, helping students or having students, working on different kind of projects with collaborators. So I'm still not there where I could say, okay, I just do only science communication. It's more like a hobby right now, which I try to really, do consistently and expand my own horizon as well with the people and the audience. So
I think that's the best thing because, that's basically also how I did it. I just did it besides for many years, and then at some point I noticed, okay, why not switch?
I don't know. Maybe you can give your take on it. I always fear a bit that you're judged because you could be standing in the lab right now, like me. I'm actually here doing this conversation with you because I feel like it's really valuable. But it could be that other people be like, hey, you should be standing at the bench and do your experiments. So
yeah, that's certainly possible. I'm aware that this was a huge factor, at least in the past. So which kept people in science from doing science communication because they thought our colleagues are gonna laugh at me for, dumbing things down and not spending time in the lab. I think it got way better. I hardly experienced this consciously, so I don't know what PIs might have thought, but usually they liked the stuff that I did.
Okay.
And then again, mostly I think, I don't know. I don't know how it's with you, but people judge all the time.
True. Yeah.
You're judged whatever you do. If you're only standing in the lab, you're judged for being a nerd or whatever.
Yeah,
don't know.
yeah. No, but that is somehow right now a bit my concerns that I'm trying to, get rid of it. Because I think right now it's well received. I was very astonished about that people really like what I do. So that really keeps on motivating, even though there are sometimes people on YouTube who
Yeah.
play the I don't like button. And I'm like,
Yeah.
why?
But then again, if someone ever explicitly says something against science communication, and you're not sitting here talking BS all the time,
Yeah.
you go way deeper than most popular science podcasts do. I think you can always argue, if there's one big takeaway message from the pandemic, then it's that if people don't appreciate science, then we're all screwed.
Yes.
There's so much value in that. And if anyone's criticizing it, most of the time, or there's a good chance that they might just be eifersüchtig.
Jealous. They're jealous.
Jealous,
Yes.
They might just be jealous, because nobody reads your paper, but people like to watch your podcast. It's the hard reality. Don't think I ever met someone outside of my own lab who said, "Hey, I liked your paper," but I'm getting getting compliments for my videos every now and then.
Okay, yeah, true. Yeah, it makes sense. That helps me also with it. And also now talking to you and listening, and that really is kind of very valuable. Thank you very much, Martin.
Thank you.
So, of course, you were doing science communication now for yourself. So what I would like to know is what are kind of rules that you stick to when you do your videos, for instance? So like how do you decide on topics? How do you think what is funny, what is not funny, especially with the background of, not hurting minorities, having the good language and so on? So how do you do it? And what are the key components you would say you always stick to?
It's hard to say. I mean, the not hurting minorities part is easy. Just don't say stupid things that hurt minorities, because why would you? But generally speaking, it's always hard to decide which topic to go for. And I noticed for me what works best is that I choose whatever is at this very moment most interesting to me. And I feel like this is what I want to now spend at least a month researching and scripting without hating myself for choosing this. And if I do, then usually what comes out as end result is something that might also be interesting for other people. I think it's tough if the thought process starts with what do I think people would enjoy watching.
Yeah.
Because that's basically the thought that everyone starts. And not everyone starts with, but if you want a video that is most generic as possible, then that's the best starting point.
Yeah, true.
And what I think is what you have to do is just, if video, for example, is the format that you're choosing, is just start, and it's gonna suck, it's gonna be terrible at the very end, and it's gonna get better hopefully from video to video. If my oldest videos, I basically deleted them because I can't watch them. I cringe myself to death. It's so bad. And it got slightly better. So
well, at least for me, I really enjoy every bit, every second, even the little sponsorships that you always add in. It's always well thought, like how to integrate the sponsorship. So I really enjoy every bit, and I take my time to watch the entire video. So I think you're doing an amazing job there, and also keeping the humor. That is important, right? So don't get too serious. So always with a laughing eye, let's say. So in that sense, I really, really enjoy it. So kudos to you.
Oh, thank you very much. Thank you very much.
So of course, I would like to know, since you're doing this now as a full-time job, right? So you are not working, so as a scientist in a lab or something. You are fully as a science communicator on your own. What is your personal goal being, being Martin on your own? What is your...
Oh, wow,
Yeah.
that's a good question. I guess I do not have a goal. My goal is, of course, that people, when they watch my videos, that they enjoy it and that they learn something. And ideally, they would be surprised how interesting this topic is. So, the trick is always I try a bit to hook them in with a little bit of humor, and then maybe they stay because not just is it funny, but they notice that this is a super interesting topic.
Yeah.
That's a bit the trick. But
so you don't have like really a plan, like a 5-year plan or something like that, what you want to achieve.
Nothing
it's
at
more
all.
like daily basis.
Yeah, the thing is, since I'm basically self-employed as a science communicator, I basically can do whatever I want. And that basically means that at this very moment, I'm doing everything I want. So the only thing I look forward in the future is maybe I want to get a bit better. So, explaining the science a bit more clearly, making it maybe a bit more entertaining and so on. And get better at it and get a bit more proficient or a bit less time wasting. That's basically it. But if I could choose what I want to do professionally, then I would say I want to make YouTube videos at home. I want to tour around with the Science Busters and to science comedy shows. I have a show also with a physicist, and I do everything that I want to do. I just want to find a good balance between doing more or doing it better.
Let's say you created already the life that you would like to have. By
a terrible thought, because then it can only go down.
No, I didn't mean it in that way. But I mean, like, how can I say it in the correct way? Let's say you have taken the right decisions that you can do whatever you want right now. Let's say it like that.
Without landing on the streets, yes.
Genau,
yes.
yeah. True. For me, for instance, I cannot imagine that for now. So I don't know why, but I just need the lab somehow. And of course, I don't generate any money with this. So I'm rather spending right now. So, okay. This is an interesting question I had in mind is, do you see differences between German and Austrian or international science communication? So I had some people from America. Their science communication is completely different, I would say. So sometimes when you see how they have to call out other people, I think here in Europe it's a bit different, I would say. But since you're a bit more into this, can you share a bit insights, what you think?
Yeah. Well, what I can say is that many other countries, they just started way earlier to, they're basically celebrating science pop stars since decades. You have figures like Neil deGrasse Tyson or Brian Cox and people like this. And this was lacking in at least Austria for a very long time. We started very early, very late. To have figures as such, and now in Germany, of course, one of the largest figures is, I guess, Martin Kim.
Yeah, true.
This chemist, she's amazing. She's such a good science communicator too.
Did you meet her at some point personally?
I met her a few times. We basically, as Science Busters, we gave her a prize at one point in Vienna, the Heinz Oberhum Award for Wissenschaftskommunikation, and we were actually singing a song together.
Nice.
We were singing a duet.
Cool.
And I was feeling so bad for her because she's actually a good singer and I'm the very opposite. Basically, I ruined the performance acoustically for everyone. And then also I met her in Switzerland because the Schweizer Freidenker gave a prize to both of us. And again, we were singing, but so I was very, I was very hopeful that after the first experience, she didn't reject it immediately.
Okay, cool. So from my standpoint, I'm just 1 year in, so I cannot talk a lot about it. But I do feel there is a difference also regarding from the educational background of the people. So sometimes, I don't know why, but it feels like that the American people are much easier to manipulate and drawn into drama than European. But it's just like I have the feeling. I don't know if this is true. So I was just thinking, like, what would be your stand on it?
Well, it's hard for me to say. I mean, people, after the last election, I think at least Austrians, they're so easy to be drawn into drama and manipulated.
We come to this section. Maybe one thing to end this part is we have the problem of misinformation and myths going on in social media and also on normal platforms. So how do you think can we tackle this problem? So you are doing already your part in taking a subject and really going down like this Granderwasser, which was really amazing. I was thinking, like, how dumb has to be the people to believe that this is true? But it turns out people are really believing in that. And these people who are running this kind of company, let's say, are also very, against scientific evidence. So how can we tackle this, that we fight against that? So what would...
Yeah. Well, the thing is, if something is very normalized, like Granderwasser, it's a kind of energized water. There's nothing in it. It's just magic, and then they say it's better for everything. I think and in Austria at least, and I think it's mostly only Austria, but maybe also a bit Germany, but mostly Austria. This is a huge thing. That 10, 20 years ago they were selling this everywhere. Every house had somehow this water energizer, not every house, but and I think it's if something is so normalized, you don't have to be dumb to choose it, because basically it's the default setting.
Okay.
You just you grow up, everyone around you says, I have this Granderanlage and everything is better, and that's the starting point. Then it takes effort to get away from that by reading into it a lot. And it's the same, with homeopathy. People are not stupid if they use homeopathy. It's just that everyone grows up getting globally, getting homeopathy. Now I forgot the question, actually.
So how we can tackle this? So of course you're doing your part already, but what kind of measurements you would like to have more implemented, maybe?
Yeah, it's hard because, there are studies that are actually, there's a meta-analysis where they actually were looking through the whole literature, which kind of methods are most effective in reducing the belief in, conspiracy theories, fake news, misinformation, and so on. And basically their conclusion is that everything that happens after people have contact with the wrong information or conspiracy theory, whatever, it's not very effective anymore. So if you just try to provide rational arguments afterwards, then it's tough. What really works is if you reach people with the good information before the bad information reaches them.
Okay.
There's something that's called inoculation. Basically, you try to teach people the structure of misinformation.
Okay.
Because sometimes they share similar structural. I think
you made a video on this, right?
I actually, wow, you're really watching my channel. I made a video.
Of course, I have to watch Martin.
And it's almost cliche, but the problem really is that misinformation on social media, it spreads so fast, because things that make you outraged. Outrage is basically the emotion that spreads the most. The more outrageous words you have in a title, the more people will click it and share it.
Yeah, I can totally understand. Yeah, and for me, it's Tell me your side. Yeah.
I just wanted to say, for me, this is almost not solvable except for technical solutions. So if you have, let's say, an AI that really is able to screen everything and maybe push the correct information a bit more than the wrong information, then maybe that's a possibility. But then again, this can be misused.
Yes. Yeah.
So easily. If Russia is using AI to select the good information from the bad, well, you can assume what kind of information will come out on top, or It's a tough problem.
It's a tough problem, and I always try to find not the answer, but maybe some, input what we can do, and also with other people who are doing science communication. And for me, it would be that maybe verifying the content of people, and also, I don't know, sharing a bit more with the audience the correct, information would be a step forward. But the problem is people are sometimes so mindless and numb. I'm not using a lot of social media, so I have to say it's, for me, something that really picks on my brain, and I'm like, I have to stop this now because it's just overloading me. But there are people who are, I don't know, can scroll endlessly, like 5 hours without knowing, and just giving a like and a heart, and that's it, and not, a critical thinking.
That's me.
I was expecting another answer here. I was thinking you were more critical. But I think, maybe there will be an AI integrate into the algorithm that makes it a bit different. I think they do a lot of changes all the time, but sometimes it affects the people that are doing good science communication. I'm not sure how is it for you right now. Do you reach your audience? Because sometimes in my case, I don't reach my people anymore.
What do you mean with reach? You mean are the videos shown?
When you upload for a reel, for instance, then sometimes I see people are commenting really weird stuff down there. So I would like to know, do you see a change right now, the people that should reach it and not reaching it, so new people are coming in, that's why you get these weird comments? Do you see shift?
Well, for me, it's a bit mysterious, this whole what is shown on social media and what not. I really, after years, I didn't manage to be able to predict which videos will be seen by many people and which not. Okay. Sometimes videos that for me they are totally banal or just so mediocre interesting, they suddenly get shared a lot and they have hundreds of thousands of views. And then videos where I think, Jesus Christ, it's the most interesting thing I've produced in years. Is getting a tiny fraction of that. I don't know if that's the question. I mean, I see that sometimes things get obviously hidden by the algorithm because there might be a keyword mentioned that they don't like.
Okay.
And that's a bit, annoying. But other than that, basically it's always the same. I'm mostly playing to my own bubble at this point. I'm aware of this. I think sometimes people send it around, this Granda video, for example, that I think many people, it's a good tool to really polarize your family if you want to send it around.
Oh, okay. Really cool. So you would say you didn't notice so many differences, or you don't just pay attention, you just upload and that's it for you?
Yeah, I'm a big fan of post and drop. So I post something and then I try to change my thoughts to the next topic.
But it's not affecting you sometimes? Like for me, for instance, I just uploaded the podcast episode with the German postdoc network coordinator. So I think it's something that we have to share. And then suddenly you see someone disliked your video. Are you already like, I don't care anymore, or does it affect you?
So I would never spend a thought on a few dislikes.
Okay.
Okay. I mean, I think that the moment you talk about a topic where it's theoretically possible to have different opinions on, it's just part of the game. As long as people don't threaten you in a way that the police has to get involved, I think it's all.
I hope that did not happen.
Internet noise.
I hope that did not happen.
Yeah, let's.
No? Oh my God. Okay. People are really
science communication around
but
the
still,
pandemic. That's
like a bit more integrity, please. Like people, please don't do that.
I wouldn't spend thoughts on dislikes.
Okay.
So maybe people see you, they think, I don't know, I hate this microphone. I had the same one and it had a defect. Fuck this microphone. And I don't know. You don't know what's happening. Also, you have to keep in mind, people are not always sober
True.
when they're at home in front of their PC. God knows what substances some people have.
Okay, I never had this
They see someone
thought.
and they hate their life, they hate everything, they just dislike and comment, and I wouldn't put too much thought into this single. If your video has 89% dislikes, then I would, maybe start thinking about the content. But besides that, it's just.
Yeah.
Actually, maybe this is a good practice that you could adopt, because when I wrote my first book, I thought, man, I've never written a book. It's probably going to be terrible. So I went to Amazon and I was looking up a science communication book that I thought is really great. It was this Dame mit Charme, actually. And then I consciously only read the 1-star reviews to make myself aware of the fact that no matter how good you do of a job, there will be people criticizing it, saying this is terrible, this doesn't deserve to be published, blah, 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 blah. Maybe then you get a more realistic perspective on what happens the moment you have some exposure on the internet.
True. For me, I try to avoid the negative bubble, because I think it's not worth giving the energy to these people. But sometimes I caught up in thinking why did this one person did this now? What the fuck is wrong with you? But then the other way is also I had 4 likes and 1 dislike, and I'm like, okay, fuck it, So I just was curious to know, because you're much, much bigger than me. So if it affects you mentally, but actually you are very straightforward and say no. I just don't. Okay,
not.
So I can learn from you. Just which helps me a lot. So that's why it's good to exchange. From your science communication part, we come a bit to a more, let's say, more serious topic. It's more like the last part now. We have now discussed you make your efforts to address, of course, persisting science myths and why is it overall important for the society. And I just recently read this article by the Community Research and Development Information Service of the European Commission. And I found this this sentence very interesting. So a well-informed public is the bedrock of a free society. Through improved science communication, we can increase the quality and effectiveness of interactions between scientists, general media and the public, and build more resilient democracies. And of course, we know now we live in a world where the political landscape, unfortunately, is shifting towards more radicalization. Without getting now too political here, but we have seen in the past elections also from Germany with the AfD and especially in the eastern side, but also now in Austria and where you are, that the skepticism for science is increasing through propagated misinformation, right? Just to name few, COVID-19 vaccines can affect women's fertility, which is completely not true. Getting COVID-19 vaccines will give you COVID, which is also not true. Or COVID-19 vaccines enter our cells and changes our DNA, which also doesn't make sense. No, it's not true. The mRNA enters the cell, but not into the nucleus where our DNA is. And importantly, it is important to make the viral proteins to actually protect you from it. And then, of course, vaccines can cause autism. There is not really this kind of link between that. And, of course, homeopathy works. No, it does not, has been shown in numerous studies. But if you have these misinformation or this myths roaming around, it poses a threat for the democracy and, of course, divides the society, which we have seen, right? So it's a big thing. Suddenly people demonstrating on streets and be like, I don't want to get vaccinated, it's bad for me, not realizing how important it was back then. So, of course, Martin, you and your mission is to educate, of course, the wider public on the common scientific myths. And what I wanted to know from you is which important elements do we need to have to deliver the overall correct information to actually strengthen our democracy again?
Yeah. You know, when we talk about the distrust in science, I think it's sometimes misleading to call it distrust in science itself, because I don't think people really distrust the science. They distrust, I think, scientific institutions. It's not just, I think, you know, there are studies on this. It's you know, we spend a lot of money on Forschungsförderung in Austria. I think one of the countries that spend most in the EU, I think even. But people, they don't protest against that. They don't say we don't want to fund research because research, we don't like research. They don't say we don't want to be part of the CERN particle accelerator because, man, science is, we don't trust it anyway. So they don't mistrust science itself. Even I would argue that many of them consider themselves scientifically knowledgeable, although they might or might not be. I think who they distrust is the scientific institutions that communicate what research shows and give directions, especially political directions. So maybe to make it a bit more graspable, they don't distrust science or the scientific method. They distrust the WHO, and they distrust the
Robert Koch Institute or
the Robert Koch Institute or the AGES in Austria. They think they're all politically motivated to the core, might have biases, whatever, and they don't trust the media that communicate science, right? They call it in Austria, they call it the Lügenpresse. So they don't think science, there's something wrong with science itself. They just think that every scientific fact that might not fit to their Bauchgefühl or to their previous idea, this is politically motivated and therefore misleading. And I think this was a massive mistake in the pandemic. At least in Austria, you had many press conferences where the politicians were standing side by side with virologists. And they were explaining stuff to the public. You know, they wanted to show competence by having a virologist on the side. But what happened is that people just saw the scientific or the people from science kind of interwoven with politics. That was the idea that, you know, that left in their head. And I'm afraid what happened is that people started to see what a scientist says more like the political opinion that a politician might say. And I think that's the even harder problem, that probably if we want to really make science education better or more relevant, is that we have to somehow build up again the trust in institutions. And that's not easy, you know, and maybe something in the institutions has to change. But also, this is something that, especially because you mentioned it before, what's right-wing parties, they really love to frame media, what we call Qualitätsmedien, and they call Lügenpresse. So the media that do the most research is what they try to frame as the most non-serious ones, most politically motivated ones. So don't trust them, trust us because we tell you the truth. And they claim they do research, but they just try to lead you astray. So yeah, this again, it's not my expertise, but
no, I mean, but so the political landscape somehow is also important to have in our mind and in the background, because as I just wanted to go on. So for instance, the FPÖ unfortunately won actually in Austria, right? They were kind of the party that was elected. But you were actually engaged in a demonstration before the Austrian election, and it was because they were denying the man-made climate change, right? So, so the motivation of you said was, I need to stand there with my science communication persona to tell the people that what they are saying is not true, right?
The thing is, I'm absolutely aware that these right-wing guys, they know full well that they're wrong. They know that there's man-made climate change. They know that the consequences will be dramatic. But they noticed that, there's a huge bubble of people who they just don't want to give up the fossil fuel car. And they will vote me if I'm, against windmills, if I'm against public transport and so it's not that they don't know. It's not like we have to be here and tell them, look, there's a thing called science out there and it shows that we're on a terrible path. And the only thing they listen to is it's not the demonstration itself. They don't care, but what they care about is votes. So the one thing we have to manage is so people stop voting for parties that, deny such fundamental claims, like this massive consensus on climate change. So the demonstration is not so much for the people in the parliament, but to say, look, there are scientists on stage, and I'm not a climate scientist. I'm a science communicator, but some people, they know me, and maybe if they say, okay, if there is such a consensus, if there are scientists and science communicators, if they're all there, maybe those right-wing extremists are wrong. maybe they're lying to us. Yeah. And so, just build up pressure. It's there's so many things that just happened when the pressure got strong enough. And it's not like people vote for their climate position. Some do, but mostly they vote for them because they're against immigration. And besides that, they try to sneak in everything else, the whole climate thing, the whole, many bad aspects that come with it. Also very democracy regarding problematic issues.
So the demonstration that you attended was more like to show the people that what they are saying is completely bullshit. And you should pay attention more to the correct information. Is this kind of
the worst thing that can happen is that if there's a problem, that is so obvious as climate change, the worst thing that can happen is that people just they just accept it and stop talking about it, stop going to demonstrations, as long as it's not solved, and it won't be solved. The question is, how big will the catastrophe be?
Yes.
So whenever there's the possibility to go to a huge demonstration, that's probably in that regard not a bad idea, because at least then it stays present. The media talk about it, and they have to, and the right-wing people then can't assume that just people will be silent anyway. I can do whatever I want.
So what are your thoughts then now? Because let's say the dust settled a bit now. How's the current situation in Austria right now? Do you know? Do you follow?
With politics?
Yes.
I follow it, of course. I don't follow it so massively. The situation is it's a terrible situation at the very end, because of course the right wing they get many positions where you don't want them, but it's an interesting position, because I don't know if that was present in Germany, but this guy Herbert Kickl is basically the leader of the very right wing party that now won. Nobody likes him. Really, nobody likes him. He was just basically, he was calling our Bundespräsident a mummy.
Yeah.
And he was just hating on everything. And everything everyone else from the other parties did was always terrible. They all suck. He's the only hero. And now nobody wants to go into a coalition with him.
Yes.
So even though they're the strongest party, everyone hates him so much in all the other parties that he won't be in the Regierung anyway.
So he will be in the opposing side, I guess.
He will be in the opposing side as the strongest party voice-wise.
And that
it
will
doesn't mean the problem is solved, right?
No,
that there will
but
be
it will
elections
make
in the
things
future.
much more difficult, right? So much more. So since they propagate kind of this misinformation against scientific topics, what do you think will be your role in the next few years, specifically when we have this kind of, also in Germany with the AfD, right? So what do you think science communicators have to be prepared for?
So I don't see my personal role in changing the political landscape besides voting and not voting assholes. But of course, it gets harder to spread all those lies, like that they're not really denying climate change, but they're basically denying everything that would be necessary to combat climate change. But that there is data showing that many of the things they say, they're just wrong. And it's probably the job of science communicators, from the field. I'm not a climate scientist, but many are. And some of them, they're good at talking and explaining things. And the more they do it, the harder it gets to
So we need to support each other.
People believe the lie. I think if you have expertise on something and someone in the parliament is talking bullshit, make a video, explain it, show the data, and maybe people spread it around, because most people didn't vote this guy. So most people would be interested in seeing how the facts are and maybe sharing them around.
Oh, okay. And that's good to know, because I already thought we lost many people, but it's just, a minority maybe just shouting out too loud. Could we say that?
I guess we lost many people, but it's not like we lost everyone.
So we need to somehow get the people back somehow. So we are trying our best. Martin, we are coming to an end, even though it was a heavy topic right now. But I always ask my guest what he or she wants to say to the audience. So the audience is yours.
Oh wow! I haven't prepared anything. Try not to lie. Try to lie as little as possible. You don't get into trouble.
So the little what's called Notlügen are fine.
I don't know. I think it's still easier in the long run if you try to get rid of those. But I assume, for practical purposes sometimes.
Yeah. So, okay. So, dear listeners, we covered a lot. So, we come to an end with our wonderful conversation with Martin. Thanks a lot for coming, for having your time. I'm really honored and grateful that you're here and shared your wisdom. So I learned a lot from Martin. And dear listeners, I hope you learned a lot and enjoyed the crossover with another colleague at some point, right? So we could call us colleague in that sense, right?
It's like a West Side Story. It's proteomics and genetics.
Yes. And also from a science communicator. Please follow him on Instagram. His Instagram is @ScienceMartin, and of course his beautiful YouTube channel called Mega. So make Europe great again.
Scheit again.
Scheit, true. Yeah, but scheit in English does not exist. So great.
Smart.
Okay, yeah. So make smart again. And of course, I don't know, but would you maybe be planning in future some collaborations? Who knows, right?
Yeah, maybe something shows up.
Yeah, I would be down for any YouTube video, and in general, also in German. So, yeah, and of course, if you enjoyed our conversation, please make sure to like it. And if you want to see more, then don't forget to follow and subscribe. And I always have a closing slogan. So I sent you the script, of course, beforehand. So I wanted to ask you if you would agree to tell the slogan with me together.
Yes, but I have to read
you have to find it first.
Yeah. Okay. Are you ready?
Yes.
Are we doing a countdown?
Yes. So 1, 2, 3. Let a little dose of honest science.
Oh, God damn it.
Okay. Okay. 1, 2, 3. Let a little dose of honest
science in your life
and brighten up your day.
Oh God, this is so hard.
Really?
I think it's latency. I think it's the latency.
On.
It's not that we're too stupid. I think it's technical issues.
Okay.
But we can. Do you want to try again?
Yes, sure, of course. 1, 2.
Or do you have a special rhythm in which you say it?
I can tell it to you. So I usually say, let a little dose of honest science in your life and brighten up your day. I usually say it like that. So, okay, let's try. Okay. Thanks for doing it.
Sure.
So, 1, 2, 3. Let a little dose of honest science in your life and brighten up your day. No problem. You want to try again? Just the last time?
Yeah, we can try one more.
I will leave it uncut like this. Okay? I will leave it uncut like this so people can enjoy.
Perfect. So we have one more. Okay.
Yes.
Okay. Maybe no, that doesn't help. Okay. Maybe we do it a bit fast so we don't stop to listen to the other one.
Okay.
Because I think that's when the latency really kills us.
Okay. So
we just,
So I just say it now and you just follow. Okay.
Yeah.
1, 2, 3. Let a little dose of honest science in your life and brighten up your day. Okay.
I think it's fair enough.
Woo.
I think we can leave that
Awesome.
one.
So thanks a lot, guys. Have fun. And please visit Martin and show some love in the comments. See you. Bye.
Thank you. Bye.
Cool.
Daily dose of honest science.